Greetings, everyone, and welcome back for more human physiology. So next on the agenda, we are going to start talking about some of the structures that you find inside of a eukaryotic cell, namely the organelles. So let's get after it here. So up until this point in the chapter, we have mostly just been talking about the cell membrane. So a plasma membrane is something that every kind of cell is going to have, both prokaryotic cells, which include most of the bacteria, and then eukaryotic cells like our cells, mammalian cells, human cells, plant cells, etc. Every cell has a plasma membrane. Well, one of the major things that separates a eukaryotic cell from a prokaryotic cell is that eukaryotes like us, our cells contain membrane-bound structures contained within. So we have a nucleus for storing DNA, we've got mitochondria, we've got Golgi apparatus, so we've got all the different types of organelles that we're going to talk about here. So you likely remember most of this from kind of your introductory biology course, hopefully. So if you look at kind of one of these pictures, one of these schematics of a eukaryotic cell that every textbook contains, you'll get a pretty general idea of kind of the location and kind of what each organelle looks like. But the thing that I'll kind of caution you here is that you don't want to read too much into this. Now, the one thing that is probably true here is that a cell usually has exactly one nucleus. Not always, and we'll kind of get into that in chapter 4, but most cells usually have exactly one nucleus, but that's about as far as it goes. So if you look at the mitochondria, for example, if you kind of take this figure literally, you would think that, okay, every cell has exactly 3 mitochondria, and that's not really true. Most cells are going to have anywhere between 1,000 and 10,000 mitochondria. So looking at a picture like this can be useful in terms of kind of looking at what the organelles look like and getting some very general information, but I do want to kind of caution you, do not read too terribly much into a picture like this. So the idea here is we're not going to spend a ton of time getting into the nitty-gritty details of each organelle. We want to just get ourselves to the point where we have a basic understanding of the function of some of the most physiologically relevant organelles, because they will come up again and again later in the semester. So one last thing that I want to kind of say before we move on to a discussion on each organelle is the distinction between these two words right here, cytoplasm and cytosol. So I will say this, it would not be an egregious sin for you to kind of mix these words up. I do it sometimes too, even though I don't mean to, but let me go ahead and tell you what these two words mean. The cytosol, that second word, literally refers to the intracellular fluid, so just the fluid portion of the inside of the cell. The cytoplasm is basically the area. It refers to not only the cytosol, but all the things in the cytosol. It would be like discussing the difference between the Atlantic Ocean and the water in the Atlantic Ocean. So one actually refers to the fluid, and then the other is really more of just an area. So for each organelle that we go through, my favorite thing to kind of do to help students get a grasp on what each organelle does is I like to kind of analogize the whole cell as kind of a big functioning town or city, and then each organelle basically has a role to play in terms of, is this City Hall, is this the post office, is this a power plant? So we're going to try to put each organelle forth as kind of filling some role in a city. So there are a lot of different organelles. We're not going to talk about every single one, but the ones that we are going to talk about, we can split up into kind of 3 different categories. The endomembrane system, which is going to include all of the kind of intracellular communication in terms of coordinating intracellular responses, synthesizing proteins and lipids, basically kind of all the day-to-day duties of a cell. Number 2, we'll talk about the energy-producing organelles. Mostly we're going to be talking about mitochondria. And then number 3, the one that we're not really going to talk about is the cytoskeleton. So things like actin and intermediate filaments and microtubules. Very important things, but not something that really I would anticipate us talking a lot about in physiological processes, so we're not going to talk about it too much. So let's start with the endomembrane system. So the main reason we're concerned about this is because these organelles that we're going to discuss here are going to underlie both protein and lipid synthesis. And we've already kind of talked about why those processes are important. Protein synthesis is going to be important for building digestive enzymes, building protein hormones like insulin and glucagon and oxytocin, lipid synthesis for building lipid hormones like estrogen and testosterone and cortisol. And then even if you want to go back to protein synthesis, cells need to be able to synthesize proteins because in many aspects, cells are made of proteins, and proteins basically do all the heavy lifting in the cell. Basically, any physiological process that we're going to talk about through the semester, if we're looking at it at the cell level, that process is going to be mediated by proteins. So we want to make sure that we have at least a general basic understanding of how protein and lipid synthesis occur and which organelles are important for this. So number one, the nucleus. So we already know the nucleus is going to be the site of DNA storage. So this is where all 46 of our chromosomes are stored. But not only that, it is also the site of DNA replication. So when a cell needs to go through the cell cycle, we are going to replicate the DNA. We go from 46 chromosomes to 92 chromosomes, and then we complete this process of mitosis to create two daughter cells that each have 46 chromosomes. This is also going to be the site of RNA synthesis, and we can talk about that in a later video when we start talking about protein synthesis itself, and then the very beginning of protein synthesis. Number two, the endoplasmic reticulum. So a lot of people kind of think of the nucleus and ER as being completely separate things, but they are actually attached to each other. So the nucleus is made up of a double-layered nuclear envelope that is made of phospholipids, and the endoplasmic reticulum is basically just a continuation of that nuclear envelope. So the membrane of the ER and the membrane of the nucleus are essentially the same thing. So the ER, depending on whether you're talking about the rough ER or the smooth ER, are going to be the sites of a lot of protein synthesis and lipid synthesis, respectively, and we can talk a little bit about that later. Ribosomes are not actually organelles, but what they are very important structures that are a combination of both protein and ribosomal RNA. These are basically little protein-making machines. If a protein is going to be synthesized in a cell, it's going to be a ribosome that does it. So ribosomes are little protein-making factories that can either be kind of freely floating around in the cytoplasm, or they can be embedded in the rough ER. The Golgi apparatus is going to be the site of protein and lipid modification, and it's going to be how we determine where cargo, once it's been made, whether it's a protein or a lipid or something else, that's going to be how we determine where that something goes. Does it leave the cell? Does it go somewhere else to be broken down if we made a mistake in making it? So the Golgi apparatus is going to be kind of like your post office there in shipping things out and determining where things are supposed to go. And then the lysosomes are going to be kind of like you could either think of it as the garbage dump or the recycling plant of the cell. It might be better to think of it as the recycling plant. So this is going to be where a lot of our hydrolytic enzymes, those enzymes that break down macromolecules through those hydrolysis reactions, the lysosome is going to be where we do a lot of that. So once we're done with a protein or done with a lipid or done with a carbohydrate or nucleic acid, we can send those things to the lysosome to be destroyed so that we can free up some monomers so that we can start brand new. So let's start with a short discussion on the nucleus and the ER. So we've already covered that the ER is a continuation of the nuclear envelope. So you can look at this top left picture and see that that is the case. So the ER is basically just this continuation of the nuclear envelope that forms this kind of labyrinth of membranous tubules and channels. And the ER comes in two different varieties. We have the rough ER, which is closest to the nucleus, and it is rough, as you can tell in both the top left and top right picture, which is an electron micrograph. It appears rough and kind of studded with little black dots, because those black dots are ribosomes. So the rough ER is rough because it contains ribosomes. And because the rough ER contains ribosomes, that is going to be where a lot of protein synthesis occurs. The smooth ER, on the other hand, does not have ribosomes and therefore is going to focus on lipid synthesis. So the smooth ER contains a lot of enzymes that modify cholesterol to make things like steroid hormones. So one of the main questions here should not be a terribly difficult question, but I just want to kind of make sure you're following along here. Why do you think it is that protein synthesis cannot take place in the smooth ER? Well, if you say, well, the smooth ER doesn't have ribosomes, and if it doesn't have ribosomes, it can't make proteins, you are absolutely correct. Now, something that this chapter in the book does not specifically mention, I don't think, but actually I do want you to kind of at least be aware of this moving forward, because this concept here will become extremely important moving forward when we start talking about something like muscle contraction. Another thing the ER does outside of just making proteins and making lipids is that it stores up a lot of calcium. So do you remember when we said that for sodium and potassium, we said that the intracellular fluid contains a lot of potassium but not much sodium? Well, if we want to add a little extra piece of information there, we could also say that the intracellular fluid does not contain hardly any calcium at all. So the intracellular fluid is going to be high in potassium, low in sodium, and very low in calcium. A big reason why there is so little calcium in the intracellular fluid, meaning the cytosol, is because the endoplasmic reticulum, through primary active transport, sucks up any calcium it finds and stores it up inside the inner part of the endoplasmic reticulum. So there's hardly any calcium in the cytosol. There's a ton of it inside the ER. And this is important because calcium, when it is released into the cytosol, can act as a trigger for a lot of different physiological events. I already mentioned muscle contraction. So the fact that we store up a whole bunch of calcium in the ER means that we can use a number of different signaling mechanisms to trigger the release of calcium from the ER by facilitated diffusion. And when we release all that calcium into the cytosol, it is going to kind of trigger a lot of very physiologically relevant events. So if you remember anything from this, just remember that one of the things that the ER does is it stores up a lot of calcium. If you can't remember anything else, then that is fine. We will cover that much later in the semester when we get into muscle contraction. So I already mentioned that I consider the Golgi apparatus to be kind of like the cell's post office, right? So if the nucleus is kind of like town hall, if that's kind of your control center, it's coordinating all the different things. If ribosomes are kind of like factories, the Golgi apparatus is definitely going to be the post office, right? So the reason why I say this is because after a protein or a lipid has been synthesized by the rough ER or the smooth ER respectively, we need to ship that protein or that lipid to where it's supposed to go. But the ER and even the nucleus don't really know where it's supposed to go. It's going to be the Golgi apparatus that makes that determination. So when a protein or a lipid has been made in the ER, it is going to bud off in a transport vesicle, just like what we saw before with exo and endocytosis. And that vesicle is going to fuse with the membranes of the Golgi apparatus. So the Golgi apparatus is this thing here that kind of looks like a stack of pancakes, right? So there are a number of enzymes in the Golgi apparatus that are going to modify that protein or that lipid. Basically, they're going to slap a zip code on that protein or on that lipid. We call these signal sequences. So it's kind of like if you take a box to the post office, you tell the person at the post office, this is where I want to send it. They are going to create a shipping label to stick on the box or onto your letter or package so that the letter carrier, whoever's going to deliver your box or your package, they know where to take it. That's essentially what the signal sequence is. It is the shipping label that tells the cell this is where the final destination for this cargo, for this protein or this lipid, this is where it's supposed to go. And then finally, lysosomes, I said, are kind of like either you could consider them the garbage dump or a recycling center, right? So lysosomes are very small organelles. They are made up of a kind of a membrane, kind of like a vesicle. But on the inside of that vesicle, they contain quite a number of different hydrolytic enzymes. And it is worth mentioning that the pH of most bodily fluids is usually somewhere around 7. So we've already said the blood plasma is about 7.4. Lysosomes are a little different. The inside of a lysosome, the pH is very low. It's going to be somewhere around 3 or 4. And that is because these hydrolytic enzymes work best at a very low pH. So what these enzymes are going to do is catalyze the hydrolysis of macromolecules. So this is the reaction in which we take apart very complex molecules like proteins, carbohydrates, triglycerides, and nucleic acids. Once those things have kind of run their course, we don't need that nucleic acid or we don't need that protein anymore. These enzymes can start breaking those bonds in between the monomers, and we can free up some nucleotides or amino acids or monosaccharides so that we can build a new protein or build a new DNA molecule or build a new polysaccharide. So the way stuff gets delivered to the lysosome is basically the same way we've been talking about before. Something that needs to be destroyed is going to be engulfed in a transport vesicle, and that transport vesicle is going to fuse with the lysosome and deliver the cargo to be destroyed there. Now, it is worth mentioning, and I think it's very interesting, that the lysosome can do more than just pick apart individual molecules. In this electron micrograph you see here at the bottom right, you actually see 2 different whole organelles in there. You actually see a whole lysosome, which I'm going to trace here with my mouse on the outside there. And inside this lysosome, you actually have a whole mitochondrion and a whole peroxisome. What we can infer from this picture is that that mitochondrion and that peroxisome are probably old. The cell probably wants to get rid of them because they're not functioning correctly anymore. So the lysosome has engulfed those 2 organelles and is going to destroy them to kind of thin out the herd and kind of cull organelles that are not functioning correctly anymore. And then it's also worth mentioning, as long as we're talking about the lysosome, back when we talked about phagocytosis, which is something a lot of immune cells in the body deal with, if a immune cell phagocytoses, say, a whole bacterium, the way that immune cell is going to destroy that bacterium is by having its lysosomes engulf the bacterium and start picking apart its membrane and proteins and what have you. So the last thing for us to discuss here in terms of the organelles is going to be the energy-related organelles. So these come in 3 different varieties. These are ones that are related in some way to energy production or somehow detoxification. So we've got chloroplasts, we've got mitochondria, and we've got peroxisomes. So I've got a strike through on the word chloroplast here, and I think you probably know why. This is a human physiology class, and we don't have chloroplasts. We don't undergo photosynthesis. That's what plants and algae and cyanobacteria do. So although photosynthesis is critical to how we live, we need photosynthesizers like plants and other things to give us the oxygen that we need and the food molecules that we need. We're not going to discuss photosynthesis in this class, and I think you're probably going to be glad for that. So what we are going to focus on mainly are the mitochondria. So we're not really going to talk about the peroxisomes, even though I did not have a strike through on that. The mitochondria are by far going to be the most important organelle that we talk about here. So mitochondria are double membrane organelles that serve as the site of cellular respiration. So you already know this is the powerhouse of the cell, even though that term kind of drives me nuts because it kind of pigeonholes mitochondria into one specific function, even though they do lots and lots and lots of different things. The reason I say that is I studied mitochondria in grad school. Kind of irritates me when people say they're the powerhouse of the cell. They do so much more than that, but that's a topic for another day. So mitochondria are double membraned organelles, meaning that they have an outer membrane and an inner membrane. So We already said this is going to be the site of cellular respiration in the cell. So we already saw back in chapter 2 kind of what cellular respiration is all about. That's going to be how our cells make the majority of our ATP that we need to power our energy-demanding processes. But as we're aware, whether cellular respiration occurs or not is going to very much depend on whether oxygen is available. So mitochondria, for most of their cellular respiration functions, are going to need oxygen. If we don't have oxygen, those processes cannot occur. So if oxygen is not available, cellular respiration will not happen. And instead, what's going to happen is glycolysis, which takes place in the cytoplasm. That can still occur, but glycolysis ordinarily is followed by the Krebs cycle and the electron transport chain, both of which take place in the mitochondria. Those are not going to occur if oxygen is not available, so if oxygen is not available, glycolysis will be followed up by fermentation. Both of these processes take place in the cytoplasm. If oxygen is available, as I was indicating, glycolysis producing pyruvate will be followed up by the Krebs cycle and then eventually the production of NADH and FADH2 that will be used to donate electrons into the electron transport chain, which we need ultimately to make lots and lots and lots of ATP. So, two different processes that can happen depending on whether whether oxygen is available. So why is it so important that we have oxygen? So it all has to do with how much ATP we are capable of producing. So when oxygen is not available, glycolysis and lactic acid fermentation are only going to be capable of producing 2 ATP molecules per glucose molecule consumed. So that may not seem like that big of a problem until you see what we do during cellular respiration. With cellular respiration, when oxygen is available, we get for every glucose molecule about 36 ATP molecules. So when oxygen is available, our cells make about 18 times as much ATP as they do when oxygen is not available. And we've already had some brief discussions on certain organs and tissues in their body that cannot tolerate not having enough energy. So your brain and your heart are perfect examples. They are very energy-demanding tissues, so it makes sense that they are going to be the ones that cannot tolerate being without oxygen. That's why strokes and heart attacks are such big deals. They need that 36 ATP per glucose because they really do require that much energy. And then the last thing that we're going to kind of touch on here before we move on to the next video and have a discussion on protein synthesis is the nucleus. We kind of briefly touched on this when we talked about the ER, but let's actually talk about the nucleus here. So the nucleus, as we know, it's where we store our chromosomes, but it's also the site of all of our RNA synthesis. That's messenger RNA, ribosomal RNA, transfer RNA, and all the other kind of more obscure RNAs like microRNAs and small nucleolar RNAs, things that you may not have heard of before, and that's okay. So I kind of already mentioned it when we looked at that kind of cookie cutter picture of an animal cell in a biology textbook. Most cells in the body are going to have exactly one nucleus, but this is not always going to be the case. Let's look at 2 exceptions to this rule. On the left here, you see some skeletal muscle fibers. Each one of these kind of long horizontal structures is a single muscle cell, a single muscle fiber. Well, these types of these types of cells actually have many, many nuclei. They don't necessarily just have one. They're actually going to have probably at least 3, maybe as many as 10 or 12. On the other extreme, if you look down here in the bottom right, red blood cells, erythrocytes, do not have nuclei because in the process of a red blood cell maturing, it basically kicks its nucleus out. You're actually looking at this process occurring in a snapshot form. On the left side, you see the red blood cell just about to kick its nucleus out through a process called nuclear extrusion. And then on the right side, you actually see the whole nucleus has been ejected out of the red blood cell. And this is because red blood cells do not need nuclei. All they have to do is focus on carrying oxygen around. They've already got all the hemoglobin they need, so they really don't need a nucleus to coordinate protein synthesis functions at all. So the nucleus is bound by a membrane structure called the nuclear envelope. We've already covered how this envelope is continuous with the ER. So the nuclear envelope is perforated with nuclear pores. It's basically kind of like Swiss cheese. So it's got these little pores that allow access between the cytoplasm, all the stuff outside the nucleus, and the nucleoplasm, which is the fluid stuff inside the nucleus. So anything can get into and out of the nucleus, provided it's small enough to make it through those nuclear pores. Now, a lot of proteins are too big to make it through the nuclear pore. So once you have a protein in the cytoplasm, it's probably not going to go into the nucleus, unless it is a relatively small protein. Now, something that is definitely small enough to get into and out of the nucleus are synthesized RNA molecules, especially messenger RNA. And this is going to be extremely important when we start talking about protein synthesis, which is going to be the topic of our next video. So, as such, we are going to cut off our discussion here. There are a lot of organelles that, if we had the time to do so, we could have talked about. We could have talked about peroxisomes and their role in oxygen detoxification. We could have talked about the microtubules and all the different components of the cytoskeleton, but I wanted us to kind of focus on the organelles that are going to be most critically important to human physiology. So that is going to do it for this video. Join us next time when we start discussing protein synthesis.